Good afternoon. Thank you for joining PHCA for today's webinar, COVID-19 Vaccine and Monoclonal Antibody Update. We apologize for any inconvenience that you may have experienced with us last week when our internet went down. Again, technology is fabulous when it works, but when it's not, it is a headache for everyone. So we appreciate your patience and understanding as we ran into difficulties last week. I'm Wendy Johnson with the Pennsylvania Healthcare Association. Before we begin, I have a few housekeeping items to cover. PHCA offers monthly webinars to members to receive updates from department staff on regulations, learn from industry experts on current trends and practices, and to gain a better understanding of practical application tools to equip you so that you may continue to provide the highest level of quality care possible. This webinar is being recorded and will be made available on the PHCA website. The webinar has been approved for 1 continuing education credit for PHCA members. Credits will be uploaded to NAB within the next 2 weeks for those of you who have provided us with your unique NAB number, and I will be sending attendance certificate. A survey will be launched at the close of the webinar. Your feedback is very important to us and our speakers, so please take a moment to complete the survey. All attendees will be in listen-only mode. However, throughout the presentation, please feel free to submit questions using your questions pane on the right hand of your screen, and our presenters will address them at the end of the presentation. Today's webinar will be presented by Adrienne Hearn, Manager of Clinical Operations Northeast, and Stephen Creasy, Director of Clinical Services, both with PharMerica Corporation. I'll now turn the webinar over to Adrienne and Stephen.
Thank you, Wendy, for the introduction, and welcome everyone to today's presentation. So, as you all probably experienced in the last year, managing the sheer volume of information in circulation around coronavirus can be quite a chore. So typically, we expect certain established groups like the FDA, CDC, and WHO as historically reputable organizations to provide both evidence-based guidance and recommendations in times like these. But what we are finding today is just that overabundance of information, which can be a barrier to good medical management in itself. Also recognize that social media allows even the most ill-informed person to be the loudest in the room, which contributes to some of the misinformation in circulation. So wading through that daunting volume of information, establishing what information is founded in fact and what sources are reputable to provide great information that you can continue to use to inform your decisions after today's presentation is really our goal. So as we move through the first 2 agenda items, we're going to be looking at the process for developing and reviewing, as well as authorizing the COVID-19 vaccines to look at it this way to help address any hesitancy concerns, as well as evaluating the CDC updates on where we stand with our immunization efforts as a country today. Now, the second issue that we found is just the rate at which the information changes. I mean, it's daily, it seems like. We got a curveball this morning from the FDA and the CDC. So we're also going to use this opportunity to visit the CDC's interim clinical considerations as they exist today, and some of the most common questions and answers that we get asked. And lastly, we do want to cover just a brief review of the monoclonal antibodies in light of the advent of vaccines and now of the variants that are out there. So let's start by setting the scene and looking at high-level data illustrating the disease burden that COVID-19 carries. So first, the CDC confirmed case numbers to date demonstrate that there's about 30.8 million confirmed cases and over 558,000 confirmed deaths to date. So bear in mind that these numbers, reaching back, approximately February of last year, are just over a year's time. So if you think of all the different therapeutic developments and interventions, like masking, social distancing, and hand washing that have been implemented in that time, that despite these best efforts to quell the outbreak, we still lost over 558,000 lives. And even operating off of the most conservative confirmed numbers, this continues to be a pretty large health burden. So if you drill down a bit further from the U.S. population at large, this graph, also from the CDC, will show you that senior populations are disproportionately have worse health outcomes due to COVID. The over 65-year age group constitutes a healthy majority of hospitalizations, while compromising nowhere near the corresponding portion of the general population. If you bring it just one step further, this long-term care COVID tracker illustrates the state-by-state breakdown of the percentage of COVID-19 deaths as a function of the total COVID-19 deaths that have occurred in the state in long-term care. So that is less than 1% of the U.S. population resides in long-term care facilities, but greater than 35% of all the deaths have been in long-term care facilities. So let's talk about the COVID-19 vaccines. So navigating this process is probably going to expose some words you may not be familiar with. We've all heard of the EUA, Emergency Use Authorization, but BLA, VRBPAC, and ACIP are all acronyms that are important even at this point of vaccine distribution to help break down the process of how the vaccines came to be authorized and to help address any hesitancy concerns. So through this discussion, we'll define the concepts of reactogenicity and immunogenicity, as well as how they apply to the general but critical concepts of safety and efficacy. And then we'll finally see how the process played out for these novel formulations that are currently authorized. So those that are listed here in green, obviously those that have been authorized. The rest are undergoing development and pending EUA or emergency use authorization submission. So let's start by differentiating what an EUA and a BLA are and what they mean for vaccine development and authorization. So EUA, emergency use authorization, has really become quite commonplace over the last year with a multitude of different drug products, tests, diagnostics, that sort of thing, getting this expedited authorization in reaction to the novel coronavirus. An example here is remdesivir and the monoclonal antibodies like bamlanivimab and the Regeneron cocktail. But what is the purpose of an EUA? When might it be used? And what does it allow authorized drug and biologic products to do? In short, an EUA functions as an expedited authorization process that bumps highly needed medical products to the front of the line, so to speak. This allows for an expedited review of any currently unapproved medical products that could be used in the case of emergency when there is no approved alternative product. So something that's gone through the traditional FDA process to exist. So it's important to note that the safety and efficacy thresholds to achieve EUA must be produced and demonstrated through clinical trials and presented to the FDA, and they make the determination of whether the demonstrated benefit outweighs the potential risk. So in this way, this process is very much in line with the standard full-fledged FDA approval process for vaccines, which is outlined there under B, the Biologics License Application. So to summarize this in the words of the former FDA Commissioner Adrienne Hearn, the standards we expect in safety and efficacy in an EUA are very similar to what we expect in a BLA. And the EUA process was actually established in the aftermath of the 9/11 terrorist attacks. And it was out of fear that the potential biologic attacks would occur, and this would allow for expedited authorization of medical treatments and vaccines to be available earlier than through the normal approval process. So the COVID vaccines that granted EUA over the past several months are not the first instance of this process actually in work. In 2005, there was an EUA for an authorized vaccine for anthrax. Now, products that are authorized under EUA do not necessarily stop there. The expectation is that these manufacturers will pursue full-fledged FDA approval through the BLA process for vaccines, which then allows manufacturers to market products through the normal channels. This is why there's no branded product name associated with any of these vaccines right now. We refer to them as their manufacturers. So a counterexample would be the antiviral that was mentioned earlier, the remdesivir, that has gone through the full process. It was first distributed under an EUA and is now independently marketed by the manufacturer as an FDA-approved product known as Veklury. So moving forward, identifying the two groups that are responsible for determining if a vaccine is authorized and approved, and making recommendations for allocation, distribution, and administration of an authorized vaccine. That's the VRBPAC, which stands for the Vaccines and Related Biologic Products Advisory Committee. It is a board of independent and unbiased members from industry experts that represent scientific communities such as immunology, virology, and epidemiology, amongst others. They are appointed by the FDA commissioner to represent these relative scientific fields with the purpose and responsibility of evaluating the phase 3 trial safety and efficacy data for proposed vaccines. Ultimately, EUA authorization is at the discretion of the FDA, but I can tell you, having sat in on one of these VRBPAC committee meetings, is their recommendations are immensely influential. The meetings are very long. They cover all the data. And the FDA determinations have not wavered from those recommendations made by VRBPAC. And something that's important to call out here is that these are publicly held meetings. They do invite opportunity for public comment and release, the in-depth summary brief in advance of the scheduled meeting. So this is all obviously in support of this process being transparent. Everything is being discussed publicly. So the VRBPAC informs the FDA, which brings us to ACIP, or ACIP, which is the Advisory Committee on Immunization Practices. So they inform the CDC on recommendations and guidance on, vaccine schedules, how to be distributed, that sort of thing. So it's a very similar makeup in terms of appointment and, committee members being pulled from public scientific fields. So what you see here is a manufacturer will, submit their investigational vaccine candidate for with phase 3 trial data to VRBPAC. They have a publicly held meeting to review the data, and at the conclusion, a vote is rendered regarding whether or not to give it emergency use authorization. The FDA will then either issue or refrain from issuing the EUA. Then, usually the next day, ACIP will review the data, as well as any public health considerations, ethical considerations, to inform the CDC on proposed guidance and recommendations, which then the jurisdictions, federal, state, and local health departments will take to form and adopt plans for vaccination with those vaccines. So when we discuss vaccine development, like all medical therapies, the end goal is really to produce a safe and effective product in that order, safe and then effective. So I've alluded to phase 3 trials in the previous slides, but what I want everyone to recognize here is how strategic and methodical the process is for any given vaccine candidate to progress through this phased approach to ensure safety and efficacy prior to being released to the public. So phase 1 and 2 trials are conducted across generally smaller number of participants to really establish that baseline safety data as well as effective dose options. And only once those favorable profiles are understood do phase 3 trials begin in a larger number of subjects. And this is really in order to gather data across a larger enough population that is diverse to draw statistically sound conclusions. These adequately powered studies are necessary for the FDA to authorize a vaccine candidate. So what the arrow points out here is that vaccines pursuing an EUA must demonstrate their promised outcomes in these large-scale trials, and for the coronavirus vaccines, it has at least 40,000 individuals. Now, a unique aspect of vaccine development during this coronavirus era has been the removal of obstacles that typically drive pharmaceutical manufacturers to approach these steps in a successive and chronological series. So that is, before investing the time and overhead resources for a phase 2 trial, a manufacturer is behooved to wait on promising results from a phase 1 before they spend any more money advancing that trial. So the key point here to kind of address the hesitancy maybe around these were rushed is that, during COVID, the intense demand and government involvement of the pre-buying of the vaccines set quantities of these potential EUA authorized vaccines has really freed up those manufacturers to pursue some of these phases in parallel rather than series, which saves a lot of time, but at the same time does not discount any of the safety or efficacy standards. And another unique aspect is that these manufacturers have now the impetus to ramp up large-scale manufacturing of these vaccines ahead of an anticipation of EA release rather than waiting to go through these different gated trials. So this is just, another reason how the vaccine production has accelerated compared to standard rollouts, where there's typically a need for marketing campaigns to drive demand prior to a vaccine's release to the market, where we already know the demand was there for the COVID-19 vaccines. So moving forward, they're talking about the safe and effective vaccine development. When you talk about vaccines, you're talking about reactogenicity and immunogenicity. So reactogenicity is just that general descriptor of the safety of a vaccine, referencing the extent to which it produces or does not produce adverse reactions, whereas immunogenicity is the indicator of general efficacy, referencing the extent to which a vaccine produces the desired immune response. So how well has this model of phase trials and methodical review processes worked in producing safe and effective vaccines? And if you remember that outside the landscape of COVID-19, this general approach is still followed, involving the clinical trial progression, the VRBPAC and FDA review, and later the ACIP and CDC recommendations. So first addressing the safety, if you recall a couple of slides ago, the candidate vaccines have to establish the favorable baseline safety profiles in the smaller phase 1 and 2 trials before advancing to the phase 3. And this is an extreme safety vetting process that has largely prevented, unfavorable vaccines from coming to market. So speaking generally, post-marketing safety issues after a product has successfully moved through this methodical process has not been a frequent concern. And it's important to note that the vast majority of vaccine adverse events occur within 30 to 45 days of vaccination. And this was actually taken into consideration under the EUA criteria that were established because they have a mandatory 8-week look-back period. So that 8 weeks or 56 days mandatory period exceeds the range in which adverse events typically present. And even for those that were authorized, those 3 that are authorized today, the look-back periods were even longer than those 8 weeks. But going even one step further, there are mechanisms in place for continued safety tracking beyond the clinical trials to provide extended periods of assessment while these authorized vaccines are being administered to the public. And that's where we are today on April 13th, with the vaccine data continuing to be aggregated and assessed and new guidance coming out. So if we talk about just briefly these safety reporting mechanisms, there's many more than the 2 here on the slide, but these are probably the most common that everyone's familiar with. So the first is Varus. It's been around, the longest. It's been around since the advent of large-scale vaccine programs, and it's the tool for reporting, recording, and tracking of different adverse events to the FDA. It's a national program managed by the CDC and the FDA, and their goal is to monitor all the vaccines licensed in the U.S. And it allows both patients and healthcare providers to report any adverse events of any severity after vaccine administration. The second tool there is VSafe, which was actually specifically developed for the COVID-19 vaccine effort. It's smartphone-based, uses text and web-based surveys, and it's really more for healthcare providers to self-report any adverse events. So staying on the safety component here, I want to review just the high-level overview of the adverse reactions, and I will touch on the Janssen, the update today, but I'm going to talk about the mRNA vaccines first, so Pfizer and Moderna. You can see that a large majority, up to 9 out of 10 patients, experience at least one local symptom, and a healthy majority experience at least one systemic side effect of vaccination. And these numbers are actually improved for Janssen's single-dose vaccine option, with closer to half of patients experiencing at least one local symptom and a similar adverse reaction rate for systemic side effects. And just so we're clear, local symptoms largely refer to those injection site reactions, such as redness, swelling, or pain, while systemic are those such as fever, fatigue, and headache. So 2 important takeaways here is that most systemic post-vaccination symptoms are mild to moderate in severity, occur relatively quickly, generally within the first 1 to 2 days, and are transient, or they resolve within several days of onset. The other severe side effects, such as anaphylaxis, did not occur more frequently due to vaccination. That was in the news and earlier, and Adrienne will touch on that in a couple slides. But post-clinical trial data across the millions of doses administered now also reinforce the safety profile that was built off the tens of thousands of trial participants. So today, there was a joint announcement from the CDC and the FDA regarding Janssen. It was with regards to 6 cases of thrombosis in women aged 18 to 49. And at this time, they've asked for a pause. And tomorrow, ACIP is actually meeting. They're calling together an emergency meeting to discuss this. And it's kind of, raised some eyebrows, obviously. It's put the halt on a lot of clinics out there and that sort of thing. But this is not an unknown side effect for the Janssen vaccine. If you actually go back to the VRBPAC briefings and materials from the meeting, it was discussed at that point in time. And then the almost 45,000 individuals in the trials there, they actually did call out there were 15 cases of thrombosis in individuals that received the vaccine. However, there were also 10 cases of thrombosis in individuals that did not receive the vaccine. They received placebo. So at the time, it was not noticed as necessarily statistically significant, but it was, they call it a numerical imbalance. And in that population, the rate was 0.05% and 0.06%, respectively. And then if you look at least the current data we have, the 6 cases, that's 6 cases within 6.8 million doses. So obviously there's a signal. It's probably the Varus system working as well as some of the other behind the scene safety analysis systems working. So I think we're all kind of waiting to see and hear the results from the ACIP meeting tomorrow. So let's talk about efficacy, the flip side, the immunogenicity. But I want to go ahead and just maybe briefly summarize some of the key takeaways around vaccine safety before we get into that. So once again, vaccine adverse events have been largely identifiable within 30-45 days. If you read the release this morning, they say continue monitoring for up to 3 weeks. So that still fits within that 30 to 45 days. And once again, there's multiple systems in place to start to monitor and to capture, and that's probably what this was. It was some sort of trigger in the safety monitoring system to pause and take a look at the data and make a decision. But moving on to the efficacy, the trial designers, they all identified slightly variable primary and secondary efficacy endpoints. So what that means is they all looked at efficacy, but with a little bit of variances in what it was. But overwhelmingly, all of these currently authorized vaccine products are effective at preventing symptomatic COVID-19 in the first place, as well as preventing disease progression to severe or critical illness in vaccinated individuals who do still unfortunately contract COVID. So as you follow the arrow to the primary efficacy endpoint, you'll want to just also account for that flu vaccine effectiveness, that's been calculated by the CDC across 10 years between 2009 and 2019 is around 44% effective. So per this standard, the FDA held at the position that the coronavirus vaccine candidates must demonstrate at least 50% efficacy for authorization purposes. So in light of the status quo, you can see how the demonstrated efficacy of these 3 vaccine candidates are still really quite impressive, especially for those mRNA. And when you discuss efficacy, we also have to recognize that the potential benefits of herd immunity or the benefit of growing immunization rates isn't just additive, but really synergistic. And what this means is that the individuals who receive any of the 3 authorized vaccines go beyond capturing the immune protection conferred that minimize the risk of contracting the infection, as well as developing the severe disease should infection occur, but it also drives the location or the local population to a critical threshold where a sufficient number of individuals possess this protection. So it's the virus's ability to propagate from host to host is hindered. So take the vaccine that's in front of you. We'll get to herd immunity. It's not necessarily always about preventing it; it's preventing the spread as well. Going back to Dr. Fauci, our go-to foremost expert on this subject. Once again, he's basically made the endorsement publicly that everyone to receive any of the vaccines that are available. As we know, allocation and supply around the country is still limited. Sometimes you don't have that choice. So you can see, just look at the efficacy, why he's saying that. To kind of, once again, address the elephant in the room with Janssen, outwardly it looks, much lower than those mRNAs at about 67%. But when you also consider some of the additional information, after 14 days, which is kind of now the fully vaccinated period, Janssen's overall efficacy is 67%, or they're just short of it for preventing a symptomatic COVID infection or moderate to severe disease. But when you look at severe critical disease vaccine efficacy, greater than a month out, the efficacy was higher at about 85%, and then 100% for preventing hospitalization. And something else to consider that's maybe not clear here on the slide is that the J&J vaccine was tested after the emergence of several virus variants. It also was at a time when COVID-19 transmission was far more widespread than during Pfizer and Moderna. And for those 2 points, it just reflects a slightly different time point in the COVID-19 pandemic for a study. So what I'm going to do here before we get out of the vaccine development, all that, to also maybe quell some hesitancy, is to talk very briefly about how these vaccines work and try to do a little bit of a middle school biology explanation here. So we all know that COVID-19 is caused by a virus that's severe acute respiratory syndrome coronavirus 2. We just call it COVID-19. But on the virus surface, there's a variety of proteins that are expressed, and those proteins are potential targets for our immune system that it can target as a potential antigen and target for destruction. So targeting those antigens is not a new concept when it comes to vaccines. It's very common. It's the platform or how those antibodies are developed. So you've probably heard the spike protein. That's what is the favorable landmark that can operate as this targeted antigen. And the key here is that this presence of the spike protein, even in the absence of a complete COVID-19 virus, can prime our immune system to mount a quick and robust response if we were to experience it in the wild. So mRNA vaccines capitalize on messenger RNA that contain instructions for making specific proteins. So for residents, I like to relate mRNA blueprints and that to your body cells to interpret or translate from, which then they're able to build structures that are detailed by those blueprints. So Pfizer and Moderna mRNA vaccines, use laboratory synthesized mRNA that instruct vaccinated host cells to make the harmless COVID-19 spike protein. The cells will then display that on their surface and cascade the immune response, which basically makes our body develop antibodies that will recognize and remember the spike protein. So anytime we're exposed to it in the future, it will make those antibodies and we'll be able to fight it off. Obviously, what's up in limbo is how long that immunity is conferred for. Common question about what happens to that mRNA. Is this gene therapy? Does it go into our DNA? And that is false. So after the function of the mRNA is complete, it is actually degraded by enzymes that are found in the cell already, and those vaccine particles are degraded, recycled, disposed of. So it's not incorporated into the DNA. It's not gene therapy. You're not a genetically modified organism after that point. Now, Janssen's vaccine falls into a different class of vaccine, which we're going to contrast here, and it's a mouthful as well. It's the viral vector replication deficient recombinant protein vaccine. So basically, a viral vector is a separate virus that is used as a vehicle to gain access into our body's host cells. I kind of think of it as a Trojan horse. Now, you might be saying that we don't want that vaccine. That why are we giving ourselves another a virus? In that, and it's really the second part of that. The explanation is the replication deficient. So it won't replicate, it won't propagate and become infectious. And then once again, the recombinant protein portion simply means that the gene for, once again, that spike protein is synthetically engineered and added to the DNA of the vector virus. So at this point, it's delivered into the cell, and unlike the mRNA vaccines which store the blueprints, the Janssen vaccine uses that DNA that houses the instructions for those blueprints within the gene to produce the spike protein, and once again, the cascade of the immune response and antibody development. So these, I believe you had the slides available to you, but these next three slides are really kind of pocket cards. And obviously the one for Janssen is going to be out of date just a little bit based on this morning's announcement, but these are just going to be quick references for you. So at this point, I'm going to go ahead and turn it over to Adrienne.
Thanks, Steve. Appreciate that. So at this point, I hope that we've established that very prescribed and methodical process that's required for vaccines to receive expedited authorization in a state of national emergency through the EUA pathway. So now I'm going to turn our attention to the progress that's been made so far in this ambitious national vaccination effort by looking at some recent updates from the CDC. Let's look at some national numbers that applies to the phase 1A individuals, the healthcare providers, and long-term care facility personnel. As of the turn of this month, there have been over 460,000 confirmed healthcare provider cases and over 1,500 healthcare provider deaths caused by COVID. Those numbers speak for themselves, but the effect is even further exacerbated by the issue of short staffing and absenteeism that quarantine, infection, illness, and of course death can cause. And I'm sure I'm speaking to the choir in that regard. In regards to the long-term care residents, I'm going to reiterate this stat, that while long-term care residents account for less than 1% of the population, they comprise over 35% of all deaths. So infection rates and mortality disproportionately affect this population, and as the graph shows, hospitalization rates greatly increase with the elder institutionalized population as well. This month's breakdown shows that there are 4 states that are in phase 1B to include only essential workers as defined by those jurisdictions. There are 9 states that have progressed to phase 1C, making COVID-19 vaccine generally available to the older adults and adults with comorbidities. 38 states are now open to the general public, including anyone over 16 and over. And all 50 states, including, or I should say, as well as Washington, D.C., have at least announced the date for vaccine distribution to open to the general public, with President Biden announcing recently that availability to all appropriate age groups must be in by April 19th. Total doses delivered as of April 10th was approximately 237 million, and it's surely grown since this live presentation. Over 183 million of those 237 million doses have been actually administered, with 7.7 million of those doses having gone into the arms in the long-term care setting, including staff and residents. 35.3% of the US population has received at least 1 dose of the mRNA series, while 27.3% of the population is considered fully vaccinated. This could mean either 2 of the mRNA doses or 1 of the recombinant vector doses. Over 7.7 million doses of the vaccine have been administered in long-term care facilities since their inclusion as phase 1A of the rollout. As important as it is to secure the supply of the vaccine for those who are most at risk, it's just as important now to overcome vaccine hesitancy for those who are working with those most at-risk individuals. The most recent data shows that close to 78% of residents have received a dose of this vaccine. However, only 38% of the long-term care staff members have received a dose of the vaccine. In the pursuit of overcoming vaccine hesitancy, let's take a look at some of the most recent safety data updates that are available. Steve has already touched on some of the breaking information regarding the Johnson & Johnson vaccine, and certainly over the next few days, there'll be more information coming on this. So we'll focus more on what we do know. By and large, some of the specific concerns that many individuals have presented based upon the early data available from tens of thousands of phase 3 participants are being put to bed now that the data is available from the millions of individuals have received vaccine post-EUA. Post-marketing data demonstrates that, just as in the clinical trials, the local injection site reactions are still the most common occurrence, and that headache, fever, fatigue, chills constitute the most commonly arising systemic adverse events. Again, these effects are mainly transient, they appear soon after administration, and they are self-resolving within several days. I do want to call attention specifically to the anaphylaxis numbers. I vividly remember emails and communications around this during the vaccine's early days, as I believe 2 individuals in Alaska suffered this severe immediate allergic response, and it made the national headlines. This is an unfortunate magnification of the negative that tends to draw media attention because that's what gets clicks. But as more doses have been administered, we've been even better estimation of anaphylaxis rates across the mRNA vaccines at 4.7 and 2.5 million cases per, 4.7 and 2.5 cases per million for Pfizer and Moderna respectively. To put that in perspective, according to the CDC, you have a 2 in 1 million chance of being struck by lightning in the next year. So walking out your door puts you at 2 in a million chance of a lightning strike with 0 improved chances of ending a pandemic, while vaccination gives you similar odds of anaphylaxis with a huge impact on your health and public health. Furthermore, all cases of vaccine-induced anaphylaxis recovered with rescue medication. And remember that vaccine immunizers are qualified and trained in recognizing and responding to these allergic issues. Impact to pregnancy has been an understandable concern for many women of childbearing age. Just hearing that you're taking something that has the name DNA or mRNA in it can raise some eyebrows. A confounding aspect to that is the ethical considerations prevented pregnancy from being an inclusion criteria for trial designs of these vaccines, and therefore subpopulation assessment of vaccinated pregnant women wasn't possible, and the official language of the EUAs does not allow for a definitive statement to be made about pregnancy. Therefore, ACIP has not jumped in with both feet to globally endorse vaccination in pregnant women, but instead just cautiously states people who are pregnant and part of a group recommended to receive the COVID-19 vaccine may choose to be vaccinated. What we can say is that the effects of COVID-19 infection can certainly be detrimental to the mother and the child, certainly with an increased risk of severe illness and death amongst expectant mothers and potential for preterm births for babies. Now that the mRNA vaccines at least have been widely distributed and administered, there is a sufficient pool of vaccinated pregnant individuals from which we can draw some conclusions, which the trials could not provide. The VSafe tracker shows that upwards of 30,000 individuals self-reported as pregnant and having received one of the 2 mRNA vaccines, and the data suggests similar local and systemic reactions following vaccination. The VSafe data, the pregnancy data, has been taken one step further with the development of a VSafe pregnancy registry, wherein the VSafe participants who report pregnancy following COVID-19 vaccination are actively contacted to enroll in a registry for purposeful follow-up. At creation of the slide deck, there were close to 2,000 enrolled pregnant individuals. Enrolled participants are then contacted once per trimester, after delivery, and then when the infant is 3 months old for follow-up. This breakdown of pregnancy-related outcomes published last month is very promising, as all currently observed outcomes showed favorable or at least no worse rates of pregnancy complications or congenital issues for the neonates. Now, the last section I want to touch on briefly are the interim clinical guidance, as up to date as I could revise to be relevant for today's discussion. Obviously, there may be some changes again coming with the Janssen vaccine, but that's to be continued. To begin, I mentioned how rapidly recommendations are changing as information comes to light. So allow me to highlight some of the updates that have been issued since the beginning of the vaccine efforts. First is something I'm sure many will be excited for, but due to the effectiveness of these vaccines in preventing the progression of the disease, fully vaccinated persons will no longer be universally required to quarantine following exposure. When we think about the mental health repercussions that isolation has, particularly in our elderly and institutionalized residents, this relaxation will be huge. Second is an update regarding allergies. We certainly don't want individuals without concrete allergies to the vaccine ingredients to skip vaccination when it's made available to them. So to this end, the CDC has clarified that only persons with a diagnosed allergy to polyethylene glycol, another mRNA vaccine component, or polysorbate have a true contraindication to the vaccine. If you're interested in knowing all the ingredients, I would encourage you to navigate to the CDC Interim Guidance Appendix C. Another point of emphasis that has come up around delayed local injection site reactions after mRNA vaccination is that it is not a contraindication or precaution for the second dose. Injection site reactions are unfortunate, but they're a normal part and anticipated adverse event. So it's really just a grin and bear it situation. The CDC has also clarified a few important scenarios and guidelines regarding the mRNA COVID-19 vaccine schedule. Continued studies around vaccine administration timing and efficacy has led to relaxation in the prescribed schedules to allow more flexibility around the 2-dose series. Specifically, there's a 4-day grace period prior to the scheduled second dose and extending the second dose window all the way out to 42 days post first dose. So this gives some much needed flexibility for recipients to manage the logistics often seen with accessing the second dose. The CDC also clarified scheduling of the COVID-19 vaccination around other vaccines, proposing a 14-day window to avoid co-administration. My personal recommendation, obviously, given the short supply and high demand circumstances around the COVID vaccine, is generally to prioritize the COVID vaccines and potentially delay others, such as the routine scheduled vaccines, as opposed to the other way around. Of course, there's a standard disclaimer. You should discuss this with your healthcare provider and make decisions on a patient-by-patient basis. Lastly, instructions were provided on what to do if the second dose provided does not match the first dose. With 3 separate vaccine formulations in circulation, the possibility can certainly arise and has arisen where a certain product was used for first vaccination and either the resident can't communicate or inform providers what dose it was or that if the formulation was available. The CDC has also offered clarification of vaccine with respect to previous COVID-19 vaccination, suspected exposure, and prior COVID treatment. Some specifics to call out: testing to assess current or prior COVID infection for the purposes of vaccine decision-making is not recommended. Also, vaccines should be administered regardless of prior COVID infection and only deferred if there's a current known infection or exposure. There is a big caveat here regarding the long-term care facility residents with known COVID exposure, that they may be vaccinated before their quarantine period is up due to the higher concern of poor health outcomes in this population and the major hurdles to get the vaccine on site. Also, patients who have received passive antibody therapy or convalescent plasma should defer their vaccination by at least 90 days to ensure immunoprotection is adequately conferred. Vaccination for individuals with underlying conditions has been a major question point that has risen, so the CDC has provided some guidance here. And summarized simply, COVID-19 vaccines can be administered to persons with underlying medical conditions who have no contraindications to the vaccination. I'm going to pass it back to Steve to talk about the monoclonal antibodies.
Thank you, Adrienne. Yeah, so the last section I'm going to cover, and I'm really going to touch on it briefly, is with regards to the monoclonal antibodies. So there are 4 monoclonal antibodies that are authorized for emergency use in adult and pediatric patients with COVID-19 who are at high risk to progressing to severe COVID-19 and or hospitalization. So these are bamlanivimab, atezolizumab, casirivimab, and imdevimab. So these are not for hospitalized patients or those that require oxygen or oxygen above their baseline. And these are the antibodies that attack the spike protein of coronavirus, just like those that would be developed by your own body after infection or vaccination. And however, unlike vaccination, they augment your immune response instead of being made part of your body, or instead of being made by your body in response as part of your humoral immunity. So your body doesn't necessarily remember. So these have really kind of waned in use with the advent of the vaccines, obviously, but coronavirus cases are still occurring, even if they are at a reduced rate. So there is obviously still a place for their therapy. So called out high risk. So what is, high risk? So for an adult, it's a pretty easy criteria to meet. So a body mass index of greater than or equal to 35, some sort of chronic kidney disease, diabetes, immunosuppressive disease, and within the long-term care population is at least 65 years of age. So for the long-term care population, it's a pretty low threshold. The pediatric side of things, there's some weight-based considerations as well as some other underlying conditions that will bump them into a high-risk category. So there was a recent update, March 24th, that was released by the FDA and with regards to variants and monoclonal antibodies, and specifically, it was around the use of the bamlanivimab, the original monoclonal antibody, being used as single therapy or monotherapy. And the concern is that an individual could be infected with one of the variants highlighted there at the bottom in the red box, that will be resistant to BAM or bamlanivimab monotherapy. So that is, treatment with BAM alone would not be effective for those variants. So what is available to pharmacies now and other locations to get is BAM is no longer orderable by itself, but you can get it in combination or atezolizumab alone, and to be used in combination with bamlanivimab. That is a combination that was approved previously on February 25th. There's still the casirivimab and imdevimab, that's the Regeneron cocktail. Or Regen COVID-19 is another term for that one. So looking a little bit just at the safety and efficacy data, as of February 10th, BLAZE-1 had enrolled over 1,000 patients. They recorded 36 events among the placebo group and 11 events among the treated group. So what BLAZE-3 and BLAZE was just randomized, double-blind, placebo-controlled studies. They were given bamlanivimab to see for treatment. So BLAZE-3 is an ongoing one. They're looking at post-exposure prophylaxis for those that enrolled in long-term care facilities. They're currently at around 1,000 individuals. So they're looking to see if the monoclonals can be given as prophylaxis. But once again, with the advent of the vaccines, that is another prophylaxis to coronavirus. So what they found and how these monoclonal antibodies are getting approved is efficacy in terms of death, preventing death. So the combined results, at least for BAMs or the BLAZE trials, was 14 deaths in the placebo group versus 0 deaths in the treated group. And they found that this occurred even if given within 10 days of viral load being able to be detected. So kind of maybe to put that into numbers. So bamlanivimab, the number needed to treat was 10 to 25 residents, and it was a 4 to 10% absolute risk reduction of death. But when you look at other things that we commonly use in patients, warfarin for AFib, in patients without a prior history of stroke, the number needed to treat to prevent 1 case of stroke with warfarin is 25. In statins, without known heart disease, to receive daily statin therapy over 5 years, the number needed to treat there is 104. And for stroke prevention, it's 154. Antihypertensive therapy for 5 years, number needed to treat to prevent 1 death is 125, and then heart attack 100 and stroke 67. So the number to treat of 10 to 25 doesn't necessarily seem as large in light of some of those others. And the last thing I want to touch on, some of the challenges that we've seen in long-term care facilities with regards to monoclonal antibodies, vascular access being one of them. That is, these are administered, via IV, via an infusion. So, necessarily having staff on site or getting staff there to place a line has been difficult. The timing, within 10 days of diagnosis, not as hard, but more so the timing with the next bullet there, the short beyond use dating. So once compounded is a very narrow window to compound and deliver and administer. So 4 to 7 hours, if it's stored at room temperature or refrigerated, you're basically 24 to 36 hours. And you have to account for the infusion time, which was, for the Regeneron products, it's an hour. The Bamlan and Atezolizumab, you can do a smaller volume much quicker now. And you can see that in the next bullet point. So infusion time, anywhere from 21 to 70 minutes, Regeneron's 1 hour. And the other kicker here is part of the EUA is the 1 hour of post-administration monitoring. That has been a challenge that has been expressed to us as well. So I know we wanted to leave some time for questions. So Wendy, I don't know if we've had any come in.
None yet, but we are open to questions. I want to thank Adrienne and Stephen for sharing this information with us today. And again, we are open for questions at this time, so please submit them using your question box on the right hand of your screen. And while we're waiting, I just want to remind everyone that the PHCA ALPC Summit is, registration is open, where we have an exciting lineup of speakers. And we are, we're planning our webinars into July, so keep an eye on our website for any new webinars that we're offering. We have a lot going on. We have probably 1 webinar a week for the next couple weeks. All right, I think that was very thorough information. I'm not sure anybody. Oh, we do have a question. Any idea on further booster vaccinations? Give me a second here. Post completion of the initial.
So that is something that is being studied by Moderna and Pfizer. I had heard or seen a couple weeks ago that they were looking out. I think Moderna, I want to say, was 60 to 70 days as a booster, but that's still one of those unknowns at this time, particularly with the variants and how that will play out as well. But at this time, there's no indication that a booster is needed. I think we're still learning about the how long the immunity is conferred for the vaccines as well. This is very much a we're all part of a very large, phase post-marketing trial type of thing right now. So more to come on that, I'm sure.
Yeah, that was a good question. Any other questions? We still have just a couple more minutes here in our hour, so we are open to questions if you have any. All right, I'm going to wrap up, seeing no more questions coming in. Again, stay on to complete the survey, if you will, please. Today's webinar has been recorded and will be made available on the PHCI website. Be sure to go and check out the scheduled webinars like I talked about. I again want to thank our speakers today, and I want to wish everyone a great remainder to your day and ask everyone to stay well.
Thank you all.
Thank you.